Just waiting for the recording to start. Okay, here we go. All right, just to confirm that you guys can hear me, I can see the presentation. Just to confirm you guys can hear me?
Yes, we can hear you.
Yes.
Perfect.
Yes.
All right, yeah, sorry, I wasn't necessarily preparing to have to do this virtually, so what we'll roll with the punches. All right, so I presented to you at the end of last lecture a video that gave us the insight as to what the principal investigator was trying to achieve with the study. And so underlying the work that was done was to try to understand what role lithium is playing in patients that are lithium responsive bipolar disorder patients. And so through this article and through the series of experiments, they identify a relationship that, or at least a role that lithium appears to play in terms of regulating phosphorylation of key proteins. And so we're going to see this explored and try to understand what particular molecular target lithium is interacting with. And so the objectives of this particular study were, as I just mentioned, to examine lithium's target in lithium-responsive bipolar disorder for molecular insight into the disease. Because most of lithium's actions have been linked to post-translational regulation of proteins rather than to transcription, a proteomics approach is undertaken to identify protein-level differences due to lithium treatment. So proteomics basically means that you're trying to track the levels of proteins and see if there's any difference in subjects. So in this context, in cells treated with lithium and not treated with lithium, and also cells that are derived from patients with lithium responsive bipolar disorder and not, right? So that's the idea of where that particular objective is centered on. Then as far as how the work really begins, it begins from a in vitro perspective using cells that are derived from patients. So these are human-induced pluripotent stem cell derived neurons. And then from there, it continues into obtaining some mechanistic validation using transgenic mice and also further exploring the primary human patient brain specimens. So as far as the approach goes, to tackle the proteomics in this study, what they did was to approach it from an unbiased differential perspective. And so what they were looking at is comparing changes in protein phosphorylation levels in these neurons from lithium-responsive bipolar disorder individuals due to lithium treatment. So what they did was they followed a SciLac mass spectrometry approach. As mass spectrometry is quite useful in being able to detect, identify proteins, but also to be able to measure the differences in levels of proteins. And then with this information, to try to contextualize what this proteomic analysis indicated and what insight can be obtained from bioinformatics studies. And then that was followed by neurocytological and behavior analyses of transgenic mice, in which an important pathway identified by the proteomic study was either eliminated or inhibited, and to try to probe the role that lithium is playing. And this was also complemented with findings with biochemical and histological assessment of primary human patient brain specimens. Right, so let's look at the first part of this approach. And so that is basically the generation of these neurons. So basically obtaining these stem cells that are then transformed into these neurons. And so they were obtained from lithium responsive and also lithium non-responsive bipolar disorder patients and patients that are unaffected by this condition, and also a patient that had Parkinson's disease, in order to have a sample from someone who had a neurologically affected non-psychiatric condition. And so let's just go to this figure here. So this was in the supplementary section of the article, which I realized it didn't actually supply to you guys, but it is available free online. So this gives us a bit of insight as to who exactly or what exactly are the patient pools that are being used in order to do these studies. And so we see in this breakdown of the where the samples are coming from, what brains correspond to people that have the bipolar condition, but also lithium responsive, non-responsive, and then what key experiments. So in this column right here, let me see if I can put my pointer on. What key experiments these particular samples, what was utilized in order to perform these experiments, and so where the corresponding set of data are placed in terms of the figures in the manuscript. So one thing that I do want to draw your attention to is this column here in the table, which is the lithium ALDA score. So basically, this is a scoring method to indicate lithium response. I'm not sure if can you guys see my presentation? Because it looks like it went off for a moment.
I can see it.
Okay. All right, good, thank you. So here, they're measuring basically the responsiveness. And so obviously this measurement will not pertain to those people that did not have the condition. But if you note here, you have these scores. And so just to give us a general gauge, a score of 7 or above indicates that that particular patient is responsive to lithium treatment. All right, so I just wanted to give us that overview of where exactly are these samples coming from, and what are the controls in order to be able to say something of significance. And so in terms of how they verified whether their protocol was sufficient given the diversity of the samples, what they saw was that there was no differences in the neuronal induction efficiency and yield from all patients. So they saw a similar expression of neuronal markers, and they produced neural progenitor cells, these NPCs. So with regards to the actual mass spectrometry experiments, so essentially what they did was they derived these cells, these neural cells, and then they, from bipolar patients, and then they subjected it to lithium treatment and compared to control. And so the idea is that you have this figure on the right that is basically the output, which is the two-dimensional differential gel electrophoresis. Basically, you're running these proteins, the complex samples, on the gel, and then all of these, when you see all of these little, and obviously sort of a mesh of different bands here. Well, essentially, then you take those proteins, you proteolyze them, you break them down to peptides, you subject them to a mass spectrometer that will do a TOF experiment. So as I mentioned to you guys previously, that is basically a fragmentation experiment. The fragmentation pattern of peptides can then be run through a database to then identify what are the proteins from which they're originating from. And so here just a representation of, and we're actually going to focus on that particular protein, this collapsin response mediator protein 2, which was identified in this part of that that fractionation of the complex protein sample that were extracted from these cells. And so what was then done was via this, using the gels to basically fractionate these samples, 26 differentially represented protein spots were identified that contained 15 distinct proteins. And so what they did was to determine the identity of these proteins. So the genes corresponding to the proteins were queried against a human gene expression database. Okay, that's a pretty standard thing to do. And so these were identified to be the ones that actually stood out. So that was the CRMP2 that I have already mentioned, also mitochondrial ribosomal protein S22, MRP-S22 for its abbreviation, and cystatin. And so these genes were the ones that were significantly increased in response to lithium and were differentially expressed in bipolar disorder compared to control brains. So what was then done was to try to see, and to an extent, to validate whether these findings correlated to other analyses, other relevant analyses. So what they did was they interrogated the Sullivan Lab Evidence Project to indicate proteins whose encoding genes might be linked to psychiatric disorders, given the bipolar disorder falls in this category. That set of studies identified CRMP2 as a gene of or protein of interest, and so that falls in line with what was observed in these in the mass spectrometry experiment. And CRMP2, as it turns out, is the only candidate protein to be encoded within the chromosome 8, so chromosomal region 8, P22 to 21, which has been linked. These are the genes that have been linked to risk factors for psychiatric disease. So this seemed to be a very promising identification. Then an ingenuity pathway analysis, IPA, and string network tool modeling was done. So it's basically a bioinformatics approach, essentially trying to see the relationship between genes to try to identify pathways that may, in which this particular protein may be involved. And so they saw that the lithium treatment of human neurons appears to modulate 4 pathways, of which that protein that was identified, the CRMP2, is part of all these. So that is the semaphorin signaling in neurons, uracil degradation 2, thymine degradation, and axonal guidance signaling. So again, all this indicates that this is a very interesting protein to follow to see if there's any relevance as to the treatment with lithium. So just to give you a bit of a context here and just some visuals for us to get a perspective of what we're actually talking about here. So this particular protein is involved in axonal guidance and also in the cytoskeletal dynamics. Phosphorylation of the protein at a key amino acid, so threonine 514, by kinases causes its dissociation from cytoskeletal protein. So this appears to be a very critical event in terms of affecting the role of that protein in terms of its contribution to cytoskeletal development. The manuscript, but I failed to look back at this detail, also alludes to another critical phosphorylation event that occurs at the amino acid S522, as sort of a precursor phosphorylation event. So IPA showed that the lithium response pathway in bipolar disorder neurons overlaps with the GSK3B. So this is the kinase that I highlighted in our previous discussion as I was introducing to you the lithium in terms of its context as a therapeutic agent. So this suggests that perhaps this particular kinase may be involved in terms of the regulation of the CRMP2 protein, and possibly that the phosphorylation of CRMP2 may be influenced by that actual kinase. So the question is, and so here I'm just providing you just kind of like a reaction to think about, is CRMP2 the primary GSK3B substrate in bipolar disorder neurons? All right, so that's the question. Is this particular kinase involved in the phosphorylation, and particularly at that critical threonine 514 amino acid? And what manifestation does that phosphorylation have in terms of activity? So here they followed the study with another type of mass spectrometry experiment. This is a SILAC labeling experiment. And so basically they were looking at differences in protein labels by SILAC labeled neurons, so neuronal cells treated and untreated with an inhibitor. So this is an inhibitor of the kinase GSK3B, because they wanted to see what would happen and there's any correlation with what is observed in this studies with those studies, or at least the results observed with lithium. And so this is just the collection of the experiments that they did. So their control being treatment with the DMSO background, was basically what was used to solubilize the inhibitor, and then treatment with 1 micromolar of inhibitor, 10 micromolar inhibitor, and then the essentially the isotopic labeling of amino acids in the media that was used to grow up these neurons. So this is basically the labels that would appear in the proteins that are going to be produced in these cells. So essentially you have your samples, you subject them to MSMS. So again, you're going to proteolyze your proteins, you're going to then identify these proteins, and you get your output of data. All right, so what was observed in this particular study? So if you look at the figures to the right, you will see how the CRMP2 phosphorylation is actually affected by the treatment with the inhibitor. So we draw our attention over here. This figure focuses on the phosphorylation of that particular amino acid, T514. And what we observe is the levels of that peptide, and at concentrations 1 micromolar and 10 micromolar, how the relative amount of that peptide, of that phosphorylation decreases, right? So you see that it basically goes to baseline at 10 micromolar treatment. This was then compared to the overall amount of protein present, so overall CRMP2, whether it's phosphorylated or not. And so as you can see here, there is a decrease to an extent, but it's not statistically significant. So essentially, the correlation with how that particular inhibitor is affecting just the expression of that protein, it does not. What it is affecting appears to be affecting that kinase is affecting the phosphorylation of that protein at that amino acid. And so further studies were done, and so these were based on this approach where they examined lithium contribution to the phosphorylation of CRMP2 in these neurons. So essentially, they examined the time course of the amount of protein, CRMP2, and the amount of its phosphorylated version at the threonine 514 during differentiation of the stem cells to the neurons and the inhibitory effect of lithium. But they also wanted to compare to see if this is actually specific to lithium, or is this an effect in general of metal ions. So remember, as I mentioned to you previously, lithium is very it's similar in terms of its transport in our body to sodium and also magnesium. So they wanted to see if some of these effects were just a general effect that was being observed. All right, so the figure to the left basically shows us the expression of the protein in the neurons, and then the expression of the corresponding phosphorylated version of the protein. And then they're using a control just to make sure that, that everything is normalized. But to our right is where we actually gauge the effect of the lithium treatment compared to sodium treatment compared to magnesium treatment. So let's draw our eyes to this, this study here. So here we're focusing on a form of the kinase that's phosphorylated. And so we can see that the bands here that correspond, that an indicator of that particular protein, we see that essentially that remains constant throughout these experiments, whether you have treatment with lithium, sodium, or magnesium. The same can be said about just the CRMP2 protein, okay, that remains constant. But what does appear to change is the amount of phosphorylated protein. And so this is observed in those treatments where lithium is present. So in this band here is where you have the corresponding 10 millimolar treatment with lithium, and then these bands where there is no difference in the expression levels, that's where we have treatment of sodium and magnesium. But then whenever you have those metals in the presence of lithium, again, you see the difference in a decrease in the expression of that phosphorylated protein, so the amount of that. So it does appear to be that lithium is correlated with the regulation of that phosphorylation site. Basically, it seems to inhibit that phosphorylation. And so this was further examined by essentially looking at other features of the inhibition that is facilitated by lithium, and in addition to the presence of the GSK3B inhibitor. All right, so here we can see this output of data where we have the expression of different proteins and in the presence and absence of lithium, concentration of 10 millimolar. Okay, so I mentioned to you in the previous slide this version of the kinase protein. So this particular version of protein, it has a phosphorylation which essentially serves to inactivate its kinase function, all right? And so what we observe here is that in the presence of 10 millimolar lithium, we see an increase in the amount of that phosphorylated protein, the inactive form, okay? Although we don't see any difference in the total amount of protein content. Then looking at the phosphorylated CRMP2 protein, we see again that the presence of the lithium results in a decrease in the amount of that phosphorylation of that protein. Okay. So additional things is that reduction in levels restores the association of that protein with tubulin. Okay, and this was something that was done in a separate study where they did a co-immunoprecipitation to verify that there is an increase in this association between these two proteins. Okay, so that would seem to be in line with the treatment with lithium was essentially restoring the function of the protein. What's not shown in this data, but is discussed in the manuscript, is that lithium increases the levels of active AKT. Okay, so the active form is this phosphorylated form, and that phosphorylated form is what it starts to phosphorylate the GSK3B. So essentially, this particular kinase appears to be what's actually phosphorylating that that other kinase, which appears to regulate the phosphorylation levels of CRP2. Relieves inhibition of protein phosphatase 2A, which is involved with the dephosphorylation of CRP2. So there's another role that the lithium appears to be playing, which is enabling that phosphatase to remove the phosphate of the threonine 514 amino acid. All right, so major findings. So the CRP2 regulation is specific to lithium among mood stabilizing medications. That lithium reduces the content of the phosphorylation at that critical amino acid in all of these cells, regardless of the patient diagnosis, was actually something that was expected. Okay, so that was a correlation that was an expected result, and it was confirmed in what they observed. But what was not expected was that the levels of this particular phosphorylated protein are significantly higher in the neurons that were obtained from the lithium-responsive bipolar disorder patients than all other patients examined. Okay, so abnormally high phosphorylated CRP2 at that threonine 514 versus the CRP2. So basically, this ratio between phosphorylation and non-phosphorylated protein appears to be disease specific. So in other words, it appears to be something that can be used as a marker. However, GSK3B phosphorylated at that site, right, that's in an activated form of that protein or that kinase, are not lower in lithium responsive bipolar disordered cells, indicative of the condition not just being associated with just that kinase. So the hypothesis being that this ratio of phosphorylation at the threonine 514 versus non-phosphorylated is a molecular hallmark of lithium-responsive bipolar disorder. And that was the working hypothesis based on these observations. Okay, so then what was done next? So essentially to then perform these neurocytological and behavioral analysis of the transgenic mice to see if this pathway is in fact affected by lithium. All right, so in vivo mice studies were performed, and so what we're looking at is data collected from wild-type mice on a mixed bulb CJ and C57BL6J background. And so we're looking at what's happening to the basically the amounts of the inactive form of the kinase and the amounts of, or at least the ratio here. So we're looking at ratios here. Phosphorylated kinase versus non-phosphorylated at that critical amino acid, and then phosphorylated CRP2 at that critical amino acid versus just protein in general. And so we're looking at treatment in the presence and absence of lithium. All right, so this data is collected from a region of the hippocampus. And so what do we observe? Well, what we observe is that in the treatment with lithium, okay, that we have an increase in the ratio of that inactive form of the kinase, okay, versus active, and then we have a decrease in the amount of the phosphorylated CRP2. And so further analysis, looking specifically sort of as the morphology of the actual neuronal cells. I mean, this can be a little bit tricky to see because you have to look pretty closely. So what are we looking at here? Well, we're looking at data collected from mice that have a knockout of the CRP2 protein. And so what was observed as far as the the profile of the mice, they appear to have normal bodies, so no indication of any differences, perhaps of mass and also of their brains, due to the absence of this particular protein. But when you actually profile the neuronal cells, here's where you do observe interesting differences. What you see is a unique dendrite aberrancy. Aberrancy means an abnormality. And so what are these abnormalities that are being seen? Okay, so if we're looking at the wild type, okay, this is a profile of the neuronal cells, and looking at the knockout, right, in terms of the visual depiction of what's going on here, you can note that there's a loss of spine density and there's an increase in bifurcation. So bifurcation basically means nodes at which the neuronal cell will basically fork. Okay, so you see these divisions that are much more profound in the knockout mice than in the wild type, right? So there are just more of those events in the actual neuronal cell. In addition, in terms of the general behavior of the mice, they were observed to be very hyperactive and over anxious. So they look to be under stress, and they also appear to be less social. Okay, and very interesting was that these mice were not responsive to lithium, right? So that appears to be an indicator, right, that that this protein is correlated with the therapeutic effect or the properties of lithium. Okay, so in wild-type mice, the CMP2 phosphorylated protein not detectable in the dendritic spines, lithium administration induces increase in spine density and area. So essentially, the lithium appeared to be helpful in order to facilitate the health of the actual cells and also the structure. So the data suggests that CMP2 function is crucial for proper dendritic branching and spine organization in vivo. All right, so that was work that was obtained with the mice. So this is more in direct in vivo work. So this other work was done with the human postmortem analyses. And so again, a lot of the work that was done and that I've shown you as far as like the obtaining the cells were collected from patients. All right, deceased patients. So as far as the patients pool that were used in the study. So the inclusion criteria for all patients included in the study was in the age range of 20 to 70, time of death, no detectable drugs or medication in the system, no other systemic medical illness. Because I imagine it's not necessarily easy guidelines to abide by as far as trying to identify patients for this particular study. And so here where we observe our data from the cortex of the bipolar disorder patients. All right, so what are we looking at here? So we're looking at unaffected dendrites versus the bipolar disorder. And so you can see the difference in terms of the profiles of these neuronal cells. And then we also see basically exploring that idea of the ratio between the phosphorylated protein versus the non-phosphorylated protein. Are we seeing the same correlations that were established in the stem cells that have been transformed into neuronal cells? So do we see the same type of relationship? And so the data suggests that excessively elevated CMP2 phosphorylated protein versus non-phosphorylated ratios are associated with diminished dendritic spine morphology, both of which are improved by lithium treatment. All right, so let's look at that directly ourselves. So that's what the article says. And so do we actually see this? And so here's the data that's presented. And so here we're looking at the ratio of the amount of phosphorylated protein versus non, right? And so we see that there is a significant amount elevated of this ratio in the bipolar disorder patients versus unaffected. But then we're looking at a decrease of the dendrite length in the bipolar disorder patients, and also a decrease in the spine density. Okay, so that's what the data indicated. And then looking at this other set of data, we're looking at then the ratio again of the phosphorylated protein, non-phosphorylated. And so we see again that elevated amount of the of this ratio in the bipolar disorder patients, but then upon treatment with lithium, we see a decrease of that ratio, basically to a point where it's statistically not different from the unaffected patients. Okay, and then we see how the spine density appears to be comparable to unaffected patients in the treatment with lithium. Right, so this falls in line with what was observed in the other studies. All right, so there was another correlation that they wanted to explore, whether there's any correlation between this lithium treatment and also the role of calcium. So diminished dendritic spine density predicts that there should be altered regulation of intracellular calcium transients with individual neurons. All right, so just basically, there was an expectation that there would be a different profile in terms of just the movement of calcium, given that calcium plays an important role in terms of the signaling within neurons. And so what they wanted to see was the signaling affected in these patients, and in particularly, so in these samples, and particularly in the presence of lithium. So neuronal function was assayed by intensity of the calcium flux. So basically, calcium flux is an indicator of activity. And so influx and efflux was steeper for lithium receptive bipolar disorder neurons, and then lithium treatment appeared to normalize the flux in these neurons. All right, so how do we see this? So if we look at this profile over here, essentially they're gauging flux by a sort of a light intensity signal. And so I don't know if you guys noticed, but there was a video that was actually they provide, so it was actually online, that shows how they actually collect some of this data. And so you see an increase in the intensity of the calcium as an indicator of the influx and then efflux, right? And so this data here, it corresponds to, in red, to the lithium responsive cells. And then here we see in blue, basically those cells that are derived from, unaffected patients. And then what happens in terms of this influx and efflux with this flux in general, with treatment in patients that are non-responsive to lithium. So we see how different it is, that particular profile. And so over here, what are we looking at is that rise and fall. So the increase and then the decrease of this flux. And so what we observe is how treatment with lithium allows for this rise and also the decay, basically to normalize, basically to reach the level of patients that are unaffected, or even patients that are not receptive to lithium treatment. Okay, so that's what the data shows here for the rise, and also very similar profile for the decay. All right, and so I just want to show you this video. That's the one that they provided, just to give us a flavor for how they basically probe some of this this flux. And so we look at the line imaging of intracellular calcium transient in these particular samples. Okay, it's a short video. All right, so let's go to where are we? Okay, so Nathaniel was the only one of the whole class who wrote a talking point for this article. I find that hard to believe, but anyways, Nathaniel. So first, whereas bipolar disorder has heritable features and developmental underpinnings, it would appear to be a disorder not of a defective gene per se, but rather of dysregulated post-translational modulation of a normally produced gene product that, although developmentally critical, plays a physiological role in all individuals throughout life. Is this research combined with previously known data enough to get more conclusions as to what byproduct may be? Seeing as the mapping of the mechanism was pretty complete, it would make sense that the byproducts may be analyzed through a list of components that react in these reaction cascades. I would say at least a preliminary insight as to a potential target of this protein. There were interesting relationships identified in terms of the lithium affecting both the kinase that was phosphorylating the CRMP2, but also the phosphorylate, the phosphorylase that was what can remove that phosphate group. So yeah, I believe there was a lot of interesting connections that were observed. I imagine since the publication that they've been doing quite a bit of work, and that's something that we can follow up just to understand the further dynamics of the role that lithium actually plays. I mean, I guess the underlying focus of this article was to try to really identify critical pathways that have a role in terms of these particular patients, to then obviously try to find a new drug target to hopefully develop a drug that's more specific for these targets. So that's that was kind of the underlying goal of this manuscript. But obviously, lithium is still something that is used for the treatment of this particular version of the condition, the bipolar receptive lithium receptive patients.
So this cramp pathway wouldn't have anything to do with the ones who aren't lithium responsive, right?
Yeah, it may not be the case. Yes, based on the other data that was shown with respect to those that were not responsive. Yeah, it may not be the case.
That would have to like make other studies to check what other possible pathways could affect that bipolar disease.
Right. I mean, it could still be very correlated with phosphorylation, just that not necessarily this part of
the pathway.
Exactly. Yeah. Any other questions or thoughts?
I have another talking point for this article is that I didn't have the time to write it down, and I didn't want it to interrupt the lecture.
Okay. You can always copy paste.
Okay.
But,
I'm trying to, but I have it on my iPad and I'm typing on the computer.
Fine.
So okay. So I'm quoting it from one of the in the article that says a qualitative, not just quantitative distinction between the lithium LIR and the LINR PBD based on an abnormally high set point for an otherwise physiological post-transitional modification of a cytoskeletal regulator invites a speculation that LINR PBD is actually a separate disease that phenocopies PPD but is unrelated. But is unrelated pathophysiological to the lithium response pathway. Right. So my question is how they were able to get to that point that the LINRPD is actually a separate disease that phenocopies the PD, but also determined that is unrelated pathophysiological to the lithium response pathway.
Yeah, so I think it's because in the if you recall the earlier proteomic studies, so I only showed you like a subset, right? So basically the treatment of the neural cells with lithium and not, but they also did it for like all the controls. And so it was the lithium responsive patients in which the CRP2 was the one that actually was one of the elevated proteins. So we don't know necessarily if probably based on their other analyses, they were able to discard the other proteins that were elevated as not being of worth pursuing, or that was just basically what they decided to focus on, given that the other types of bioinformatic data suggest that this protein was of interest, right? So some of those studies were the indicators that this particular protein may not be involved with that other manifestation of bipolar disorder. So in the last lecture, I kind of generalized like there are 2 or 3 types of bipolar conditions, but in reality, there are a lot of like conditions that mimic what is a profile for bipolar disorder, but that don't necessarily have all the same. I would say you see a lot of the same physiological conditions, but they not actually have the same origin. And so here, I think that's what they were trying to indicate with at least comparing those patients and what they were able to identify from the corresponding proteomics analysis as that this particular path or this particular protein is part of the, I guess, the dynamics of that or even the manifestation of that particular bipolar disorder condition. So yeah, so that's I think where they're essentially getting that, or at least they're drawing that conclusion. Any other thoughts? Thank you. Yeah. Any other thoughts, questions?
I had a question. I didn't write the talking points for this article because I did the synopsis, but still, I wanted to comment that when they measured the radio of the kinase, the GSK3 beta, the radio phosphorylated and not phosphorylated, it was seen that the baseline radio for affected patients and not affected patients were similar, and I would expect for it to be lower because well, it would seem that then it affected the phosphorylation of the CRMP2. And I wanted to know why did this happen? I know that they mentioned that maybe it's because something else is involved in why the ratio of CRMP2 phosphorylated is higher in bipolar patients, but why didn't this happen? Like I would have expected for this radio to be lower.
Right. So I think what's going on here is in general, the phosphorylation is something that happens. So there's like a sort of a balance of how much the proteins get turned on and off, because that's basically what that is a control of the activity of these proteins in this context by phosphorylation. So I think that what they're just seeing in this particular case is that this particular phosphorylation is basically completely off. And so with the lithium responsive patients. So I think that's the main take-home message, that this processes are normal, like that you would expect to see some amount of phosphorylation, except that in this particular disease pool, we're seeing that this ratio is quite elevated against the, I guess, healthy patients, if you want to put it from that perspective, or not even necessarily healthy patients. So I think that's what we're supposed to focus on, if that makes any sense.
Yes. Okay. Thank you.
Professor?
Yeah, go ahead.
I already did the synopsis and the talking points. I put it on the paper.
Yeah.
I wasn't so sure how to put it, but I can say it. But I don't know if I can say it in verbally instead of on the.
Yeah, go ahead.
My talking point about the paper is that it's so well known that at least with lithium, it's useful with in neurology study in terms of helping the patients with neurodegenerative diseases to try to minimize the effects and to try to heal and recover them. And yet it's interesting that it is not so well known how the mechanism of how lithium is actually done. I mean, with the paper the article that I read, there were some people that were debating whether the mechanism is one way, yet they say the other way. But most of the paper overall generally concludes that the way how it's done for to inhibit, I don't remember right now, the GSK3. There was another one. I don't remember that one well, but those two are the target points, and that inhibits it so that would cause a way to proceed a therapeutic healing to the person with that process.
Right. So I would say something that as chemists we see like the levels of these proteins and the phosphorylation changes. What we're not getting, at least it was not so clear at the end from reading this, and then obviously I would have to follow up with this particular line of work, is lithium actually binding somewhere that's actually facilitating this? There is last lecture I mentioned that lithium can bind at very key, catalytic sites of these kinases, these phosphorylases that where magnesium would normally bind. But here in this particular study, that is something that that detail, that molecular detail is missing. So it's not so clear the direct interaction. We see the effect that it's causing, but we don't quite get to that level of point. At least I didn't see that detail in the discussion. All right, guys. So we're gonna move on to then our next discussion. Let me go ahead and close this. Switch over here. Am I still sharing? I'm still sharing.
Yeah.
All right, good. All right, so we're going to revisit, and because this is going to be a real focal point for our discussions of the next set of lectures, but also a discussion of our review article that we will meet next week to focus on. And so this idea of the role of metals and also immunity. And so we're going to talk a bit about metals and immune-related diseases, just to give us a little bit of insight into what role metals can play. And we already touched upon this briefly when we were discussing nutritional immunity. So far, what we have discussed is this idea of balancing metals. With the story of iron, we talked a little bit about how there are those molecular mechanisms in place for maintaining the homeostasis of this metal and regulating its bioactivity. We looked at the example of a loss of homeostatic biodistribution of certain metals in Alzheimer's disease patients. And so we discussed a little bit about approaches that people have taken to try to develop therapies for this particular disease. Again, we discussed this idea of nutritional immunity and how our body responds to pathogenic agents by basically perturbing our essential metals in a defensive manner. And so in this last example, when we're looking at lithium, we're looking at a situation where, in that particular case, a metal that is a little bit of a question mark as whether it's an essential metal. I mean, there's indicators that suggest that it is, but certainly the role of that metal in terms of balancing protein phosphorylation and a potential therapeutic effect from that process. So what we're going to look at here is a little bit more about immunity. And so I just want to give you just an overall perspective. And so these are things that you probably saw in biochemistry. So I'm just going to just review a few key points here. So the human body has two lines of defense: the innate immune system, which is basically a rapid response to molecular features that are present in pathogens or on pathogens, and the adaptive immune system, which is responds to the specific features present on a given pathogen, right? So this is sort of the a response that your body will develop. Both work to eliminate or to neutralize. That's the terminology that we're hearing a lot as we hear about the COVID-19 pandemic. Those pathogens. So here's just a picture, kind of giving us an overview of this innate immunity versus the adaptive immunity. Okay. And so we're going to talk a little bit about the role of certain cells in facilitating this defense mechanism. But what are the challenges that are inherent to identification of pathogens? Because I'm bringing this up because I'm going to discuss autoimmunity, okay, and why it's important to understand those type of conditions that can happen. So the challenges: to produce a system of receptors diverse enough to recognize a wide array of potential pathogens, right? So you can imagine that can be quite complex, especially given that we have our own internal molecules that our body has to be able to, process and distinguish from something that is foreign. So to distinguish invaders and their disease-causing products from the organism's own or the host's own products. So this idea of a self versus non-self recognition. So the innate immunity is an evolutionary ancient defense system found at least in some form in all multicellular plants and animals. And it's basically the first line of defense against foreign pathogens. The idea being that you're trying to recognize through this mechanism common features of invading organisms. So common features, it would be a little more specific here, some molecular details. So well-conserved, for instance, specific glycolipids or nucleic acids that are not present in host organism. So the same idea is what people, when they try to develop these antimicrobial agents, is you try to develop something that will target something that is distinguishable in a pathogenic agent, and that's not something that you would find in humans. So, components of the innate system include the epithelial lining that surround host cells, specialized cells called phagocytes that can ingest and destroy pathogens. Right, so these toll-like receptors are actually really important in this line of defense. These proteins are among different family of receptors that are expressed on antigen-presenting sentinel cells. So when I'm mentioning this, I mean macrophages, dendritic cells. These are immune cells, okay? And that these receptors can recognize specific features present in most pathogens. So you will find these receptors on cell membranes or internal organelles, in both, okay? And these are some of the common features of the toll-like receptors. So one of them is this large extracellular domain with 18 to 27 leucine-rich repeats, okay? So each with 20 to 30 residues, of which 6 of them are leucine. And then you have this feature here where it's capped by a cysteine-rich domain. You have the single transmembrane telix, and then you have an intracellular signaling domain, which is basically a docking site for other proteins. So here's a depiction of the leucine-rich repeat. Okay, so this is just the repeat, where you have the 6 leucines present in each of these repeats. Okay, so the leucine-rich repeat units come together to form a central parallel beta sheet that curls to form a concave structure. So you have this this beta sheet arrangement and this structure, this curved structure that forms. So essentially, within each of these repeats, you have the leucines that are creating a hydrophobic core. So something that's a very critical thing about the function of these type of receptors is what are known as the pathogen-associated molecular pattern, or the PAMPs. PAMPs are the specific molecular characteristics that each toll-like receptor targets on invading organisms. So that's what they recognize, the molecular feature. Typically a critical component of the pathogen that cannot be readily mutated because that mutation of that component would affect pathogen function. So that's, our body has evolved this sophisticated system to target those essential molecular features of these pathogens that they simply cannot evolve, otherwise it would actually affect survival. So this table provides just an overview of the different toll-like receptors, where they're localized, what PAMP, so what molecular feature is recognized, and the source of the PAMP, so what microbe is being detected here, or what's the source of that actual molecular feature. And so what we have here is a recognition of a PAMP by a toll-like receptor. Okay, so we're seeing just a structure of that particular interaction. So then we also have the adaptive immunity. So this comprises 2 parallel interrelated systems, the humoral and also the cellular immune responses. The humoral immune response, here you have soluble proteins called antibodies. Typically, we also refer to them as immunoglobulins. They're very abundant in our blood. Function as recognition elements that bind to foreign molecules and serve as markers signaling foreign invasion. So basically, they bind to a foreign agent, and that's a signal that this thing needs to be removed. It's not part of the system. Secreted by plasma cells, which are derived from B lymphocytes, or B cells. So a foreign macromolecule that binds selectively to an antibody is called an antigen. The specific antigen is not for the entire macromolecular antigen, but for a particular site on the antigen called the epitope. Right? So you're just basically recognizing one part of the whole agent here. So each beta cell, B cell, produces just one type of antibody that can recognize a single epitope. The immune system generates more than 10 to the 8 different antibody proteins. So here, this table presents you properties of immunoglobulin classes. So these are the antibodies, the concentration that you find in serum, the general masses of these, and so basically the structural details of these immunoglobulins. Okay, so they also have the cellular immune response. So this consists of cytotoxic T lymphocytes, so killer T cells that destroy cells that have been invaded by a pathogen via identification of markers on the cells. Right, so a cell that's invaded, then it's labeled, and that label is what's recognized by these T cells. The cellular immune response is mediated by specific receptors that express on the surface of T cells. Because intracellular pathogens do not leave markings on the exterior of infected cells, vertebrates have evolved a mechanism to mark the exterior of cells with a sample of interior contents, both self and foreign. So let's have a little gauge of what this actually looks like. All right, so some of the internal proteins are broken into peptides, which are then bound to a complex of integral membrane proteins encoded by the major histocompatibility complex. All right, so what we're seeing here is just basically a depiction of a T cell, okay, that is interacting with the major histocompatibility complex of the MHC. And so this is a contact by recognition of something that was that's basically coming from the foreign agent within the cell. So that's what the indication of a proteolytic event that then takes that a piece of it, a peptide in this case, labeled, and so that's an indicator that there's something foreign here. And so you see this interaction between the receptor and the MHC. All right. So the immune system generates more than 10 to the 12 T cell receptors. Helper T cell, or helper T lymphocytes, helper T cells, for abbreviation, contribute to both the humoral and also the cellular immune responses by stimulating the differentiation and proliferation of B cells and T cells. All right, so the T cells are subjected to a process of positive and negative selection in the thymus. So I just wanted to give you this visual of where the thymus gland is located. And so that is situated above the heart. And so when you're going through this maturation process, T cells that are produced in this gland, they are subjective to what is known as a positive selection. So those cells for which the T cell receptor can bind with reasonable affinity either to class 1 or class 2 MHC molecules survive the selection. Anything that doesn't have an affinity for the MHC molecules will basically be killed. So they will undergo apoptosis, which is a programmed cell death. So after this process, then comes negative selection, where you have here T cells that bind with high affinity to MHC complexes bound to self-peptides. Okay, self-peptides meaning peptides from the actual, from the body, expressed on the surfaces of antigen-presenting cells in the thymus undergo apoptosis. So anything that has a moderate affinity will complete maturation. So you want something that has sufficient affinity but can allow for dissociation. Okay? So anything that's just too high of an affinity to the MHC complexes with the self-peptides will get eliminated. This process is actually very important. And so this leads into what I was alluding to, which is autoimmunity or autoimmune diseases. So that results from the generation of immune responses to self-antigens. So despite the efficiency of the thymic selection, so the thymic selection again is that positive selection followed by the negative selection, failures do occur. So autoimmune diseases include such conditions as insulin-dependent diabetes, multiple sclerosis, and rheumatoid arthritis. And so result in damage to selective tissue that express the antigens. So in many cases, it's not very clear how these autoimmunity conditions actually arise. But possibilities of what could be, a source of origin. So in some cases, infectious organisms such as bacteria and viruses may play a role. Infection may lead to the generation of antibodies and T cells that react with different epitopes from the infectious organism. If one of the antigens closely resembles a self-antigen, that can then produce an autoimmune response just because of that approximation to a self-antigen. So that could potentially be a source of autoimmune disease that's actually triggered by, by a virus or a bacteria. I will say, just to mention briefly, there's more interest being explored about how the microbiome may actually play a role in terms of autoimmunity. So the contributions from those particular organs that are part of the body, and possibly some amount of loss of the homeostatic control of these organisms that could lead to autoimmunity. All right, so let's explore a little bit about mechanisms of heavy metal-induced autoimmunity and how this manifests itself. So here I refer to this article for some of the information. This is actually not coming from the main textbook that we've been using for the course. So as you can imagine, and we've already looked at this to an extent, that metals can play both beneficial and detrimental roles with respect to the immune system. But metals themselves can be seen as invaders, right? Just like if you think about pathogenic agents, metals can basically be perceived as something that's foreign, right? Especially if they're not something that is essential. So several transition metals, namely mercury, silver, gold, have the capacity to induce changes in cells of the immune system. Mercury is one of the major ones to trigger an autoimmune effect. So let's talk a bit about mercury. So when we were talking about hard, soft, acid-base chemistry, well, mercury is one of those metals in the metal ion form that falls into the soft acid category. So that's going to be really important when we discuss some of the manifestation of autoimmunity that comes from mercury in the body. All right, so mercury is prevalent in the environment. It can be a major pollutant. It is found in a number of medical treatments, so found in dental amalgams, cosmetics, vaccine preparations, also found in preservatives and fumigants. So it can get into the body, and it's actually used for a number of things that we depend on. Well, exposure to mercury, so what are those indicators to suggest this? Well, some of these are the elevation of T cells, also elevation of the antibody immunoglobulin IgE. Some people produce anti-nucleolar antibodies. So the nucleolus is the largest structure in the nucleus of eukaryotic cells, which is important for ribosome biogenesis. So these antibodies basically form due to the recognition of a marked nucleolus as foreign. So something happened, something biotransformed this nucleolus, so that now it's recognized as being something that is actually foreign. And so in order to gain some insight as to the metal-induced autoimmunity, studies have been done with rodents, those that are susceptible to metal-induced autoimmunity. Okay. So studying genetically susceptible rodent models allows insight into how metals can induce an autoimmunity. And this is important to give some insight as to how certain, given that many metallic materials are introduced into bodies, especially in prosthetics. So one particular animal model that is susceptible is the brown Norway rats. So they exhibit autoimmunity when dosed with subtoxic amounts of mercury chloride. So what that means is basically below a level that would kill them, right? But there's still associated conditions, and so in this case, an autoimmunity that forms. So similar profile is seen in these particular rat models. There's a production of antibodies, and so you see all these different types of antibodies that are produced against their own biomolecules, so DNA, phospholipids, and so forth. Also, polyclonal activation of B and T cells, and also a renal immune complex deposition resulting in and basically this inflammation of kidneys. But it turns out that depending on the exposure, the rats can become resistant to mercury. To mercury in the form of 2+. Okay, another animal model, rodent model that is susceptible is lupus-prone mice, and these have been shown to exhibit a similar profile as the susceptible rats. The brown Norway rats is an example. For instance, autoantibodies form. The ones that form appear to be restricted to the nucleolus. And so the main target of these autoantibodies is fibrillin, which is a 34 kilodalton ribonucleoprotein protein that is associated with several small nucleolar RNAs. Right, so what are some of the proposed mechanisms of the metal-induced autoimmunity? Well, as given, autoimmune response is part of the adaptive immune system. Okay, so that's just a general statement. But mercury can affect the expression of various genes in immune cells. So in that vein, it can affect the proper behavior of the immune system. Though subtoxic dosages are administered, there may be localized toxicity. So what that means is that, and I've mentioned this a few times, this idea of compartmentalization within cells can lead to compartmentalized elevated amounts of a given species. So perhaps some of the manifestation of the effect of mercury may be due to localized elevated concentration that induces toxicity and cell death. And so mercury-induced cell death results in a 19 kilodalton cleavage fragment of the fibrillary protein. Another really important proposed mechanism is the interaction. So this is now we're talking about the metal binding property of this metal. So mercury 1, mercury 2, in addition to those other metals that I mentioned at the very beginning of the section, silver, gold. Well, they are soft Lewis acids, and so they have a strong affinity for thiol groups, right, being soft bases. So what is observed is that the ability of mercury to be able to bind to a number of these soft, localities within proteins, within biomolecules. So for instance, this small molecule, glutathione, is a very important molecule in terms of regulating redox within our cellular environments. This is a molecule that is abundant, millimolar amounts in our cells, right? And so it is the most abundant source of intracellular thiol. And this is basically a tripeptide, so that it consists of 3 amino acids, glutamate, cysteine, and glycine, where actually the glutamate is forms an amide bond, but is actually forming through the side group and not through the terminal carboxylate. But so basically, this is a small but very important molecule in terms of redox regulation, and it has this really significant thiol group, and that thiol group plays a big role in terms of when we were talking about iron, it actually plays a big role in terms of the labile iron pool that transiently forms within cells. Okay, so binding to the thiol group of glutathione may cause a redox imbalance, given the role of glutathione in that process. So perhaps coordinating mercury will then inhibit its function. What has been observed is that it inhibits glutathione's regulation of concatenating A-mediated induction of cytokine interferon gamma. So in general, cytokines are a family of proteins that bind to specific receptors to stimulate B and T cell growth and differentiation. Right? So this is an important process. That's actually part of an immune response. What's also observed is that treatment with exposure to mercury, exposure to mercury can cause a shift in the expression profile of cytokines. So this may be correlated with binding to glutathione and also possibly binding to just thiol groups in general. Also, as it has been observed, is that mercury exposure can dramatically worsen other autoimmune manifestations. Right, so I wanted to introduce that just to give you a perspective of how certain metals can lead to an autoimmunity given their metal binding properties, but also other indirect consequences of what they can contribute to in terms of the manifestation of those particular diseases. So what I want to focus on here, and is actually is going to touch upon this a little bit about autoimmunity, but also the role that the metals can play in treatment of an autoimmune disease. So we're going to see a little bit about how a certain metal can play this role of treatment, but also can play the role of basically increasing the actual disease condition. Right. So that's sort of the unfortunate side effect of some of these metals in terms of this particular application. So here, focusing on rheumatoid arthritis as an example of an autoimmune disease. So this is a disease in which the body attacks the lining in the joints and the fluid in between. And so what is observed physiologically is that is an erosion of opposing bones, and also can lead to severe impairment of movement. And generally speaking, what's done to try to treat this condition is the use of painkillers, and also the use of disease-modifying arthritis rheumatoid drugs, so abbreviated as DMARD. So these are immunosuppressive drugs. Well, chrysotherapy was a therapy that was developed from gold-based compounds. And so these particular gold compounds were shown to have an actual therapeutic effect on patients that were suffering from rheumatoid arthritis. So the phrase chrysotherapy was coined by the French physician Jacques Rossier. So the original generation of the gold compounds were gold 1 thiolates. So if you guys can recall, gold 1, very soft, right? And so these compounds, in terms of the of their coordination chemistry, they make sense. They fall in line with what you would expect. They form compounds with these thiol groups. And so you have sogenol, you have cyanochrysin, you have myochrysin. The main drug in this family of drugs is Aranafin. So Aranafin was actually approved for the treatment of rheumatoid arthritis. It entered into the drug market in 1985, although today is rarely used for this purpose. But this was definitely a lead drug in its time. So it's an orally administered drug, and so here we have the composition of this drug. So it's a gold 1 linear compound where you have this thiol glucose that is acylated. Okay, so the coordination through the thiol group, and then you have this triethylphosphine, also a soft Lewis base that's coordinated to the gold center. So you have this linear 2-coordinate compound. It reduced kidney toxicity relative to other gold thiolates. One of the reasons why it really advanced to the market. And part of this was due to less excretion via kidneys and reduced overall gold retention. This idea of retention actually is a major factor here in terms of the overall therapeutic effect of this particular drug, or of gold in general. All right, so talking about this idea of retention, well, let's talk a little bit about the biodistribution of Aranafin. Well, it turns out that this drug is not very water soluble, and the effects of the drug are really slow to become apparent. In some cases, it was observed that it could even take up to a year for any effect to be observed. And that in part was due to a slow accumulation of gold throughout the body. So here what I'm going to present to you is just an overview of the clearance of the drug in the body. So what I'm showing to you is isotopic labels of a study that was done in which they used isotopic label form of the actual drug. We have S35, gold 195, and phosphorus 32. So basically you're tracking the fate of those corresponding atoms. So you're tracking the gold and you're tracking the coordination points of the two ligands. So it turns out that in terms of the excretion from the body, the sulfur component of this molecule, well, that has a half-life of 16 hours. The phosphine portion of this has a half-life of 8 hours, so it's even lower, okay? But then the gold portion of the drug has a half-life of 20 days. So clearly, this drug is not necessarily remaining intact, right? There's a clear difference in terms of the profile of the components of this drug, where the gold itself stays much longer in the system. And so how do we get some insight into this? Well, some of the insight is afforded by getting some idea about the in vivo speciation. And so if we're thinking about this as an orally delivered drug, then we have to consider the speciation within an environment that approximates metabolism, that approximates the gut. And so the pH, to mimic the pH of the stomach, a study was done to look at the degradation of the compound at a pH of 1.6. So this was a study that was done by Stop Flow Kinetics using a UV-Vis detector, basically studying the absorbance of the compound. We've talked about UV-Vis absorbance of metal complexes before. All right, so this was done in a background of 0.5 molar chloride. So a pH of 1.6. So essentially, this is the model that they propose in terms of the speciation, where you have this 2-coordinate compound in the presence of HCl, so pH 1.6, results in initially a coordination of the chloride to the gold center, but then very, very quickly a dissociation of the thioglucose. And so essentially that the coordination of the chloride leads to the dissociation of the thioglucose. That occurs at a half-life of 0.01 seconds. So you can see that this compound in the acidic environment approximating the digestion in the stomach, it rapidly degrades. And so it turns out that 25% of the oral dose of this compound is absorbed from the gastrointestinal tract. So it's only 1/4 of the drug dosage actually gets absorbed into the gastrointestinal tract. So that amount then is distributed through the body. So this process of the breakdown, but that's basically giving you the indication of how quickly it will get processed in the stomach, and then could possibly lead to why part of the thioglucose is excreted from the body, right, without even actually entering into the bloodstream. All right, everyone, I'm going to stop here, and we'll continue from our discussion from this point to look at the profile of the speciation in this drug and to get some insight as to how the drug actually manifests a therapeutic effect. Everyone, so I will see you next time. Claudia, if you have a moment to stay behind.
Yeah. Right here.
Thank you, Professor.
You're welcome. So I'm trying to let me stop the recording.